It all starts with a vision, the foresight to see the possibilities ahead, and the brilliant ingenuity to bring it to life, and the skill to make the impossible possible. It is understanding your role and using it to shape the way forward. It is to recognize the freedom of creativity. It's having the ability to be bold, yet stay relevant through the years by creating forms that are meaningful and lasting. Built to beautify your life, built for you to thrive. It is this spirit of perfection and wanting better that fuels our purpose at Nations Trust Bank. A philosophy that drives us to be more than a bank and recognize our ability to shape life's present and future by building relationships that endure. Shaped the right way, shaped for better. Nations Trust Bank. Trust in better.
We have two generations coming together today. We have Hiranya, who's a 33-year-old young scientist who's been exploring freshwater fishes since his childhood. He gave up a career in medicine to study zoology, securing a first class in his BSc at Pethiyagoda, and going on to do his MPhil. He has discovered and named 7 new species and 3 new genera of fishes. And despite his youth, he's the author of more than 30 international journals. He also went on to rediscover 2 species which were long thought to be extinct. He's presently in his final year of his PhD program at the University of Bern in Switzerland. He's joining us online via Zoom, so he'll basically take up Q&A. The presentation, as you know, was prepared by Rohan and him together, and Rohan will do the presentation. Rohan, he needs no explanation, but for courtesy's sake, Rohan, we need to give a little introduction. He's widely known as the David Attenborough of Sri Lanka. His remarkable ability to bring science and nature alive to people of all ages. He first gained national recognition in 1991 with his landmark book, Freshwater Fishes of Sri Lanka, the first comprehensive guide to feature color photographs of every known species at that time. Since then, he has authored and co-authored numerous books and scientific papers, including Ecology and Biography of Sri Lanka. That's a book that we just have another 3 copies left, I'm told. His work has led to discovery and description of hundreds of new species, and there are so many species actually named after him as well in Sri Lanka, making him one of the most influential voices in Asian biodiversity research and conservation. Rohan's contributions have been recognized internationally. He's a recipient of the prestigious Lenin Medal for Zoology, Rolex Award for Enterprise, and a Fellow of National Academy of Sciences. At home, he has long been associated with the Wildlife and Nature Protection Society as a life member, and also a former editor of our Laurie's, the Journal of the WNPS. With his deep knowledge, storytelling gift, and lifelong dedication to protecting Sri Lanka's natural heritage, Rohan continues to inspire generations of scientists, students, and nature lovers. Over to you, Rohan.
Good evening, everyone. It's great to see such attendance on a day that's as bad weathered as today. When I wrote Freshwater Fishes of Sri Lanka 35 years ago, as Spencer said, I tried to bring together the then available information about all these species together with color photographs. And that was the first time that any group of Sri Lankan vertebrates had been treated in that way, as a full color, intimate book bringing together a fauna with so much endemism in our country. That got the study of freshwater fishes and enthusiasm going, rather like Sarath Kotagama's book published 3 years later, Sri Lanka Kurullo, sold 20,000 copies and took bird watching to the village. Amongst the generation of young scientists that then started studying our fishes was Hiranya, who, from the time he was in school, was corresponding with me about freshwater fishes. And for the past 10 years, he has been studying them intensively. His interest was mainly on the genetics of fishes, where I had been talking about the old-fashioned morphology and taxonomy. He wanted to turn a new page. And we laid a framework for that, a context for that, in this book we published in 2021. Now he's doing his PhD in Bern, problem with the slide, but on these minute fishes, the world's smallest fish group, Pediocypris. And he's trying to find out how the genome of these minute fishes varies, because this family, the order to which Pediocypris belongs, the Cypriniformes, is an order that also has some of the world's largest freshwater fishes. This one, Catalocarpio, for example, is 15 million times the size of a Pediocypris. Hiranya's job is to try and find out how the genomics of these two species and the range of species in between varies from an evolutionary standpoint. Now, I think the freshwater fishes of Sri Lanka are now well known enough that I don't want to give you a slideshow about those fish. So we thought we'd approach this from a new perspective, because I want to emphasize something that we don't pay enough attention to when we think about conservation in Sri Lanka. And that is evolution. So before I get started, we have to first understand a few basic things about evolution, because not everyone might be familiar with them. And for this, we're going to have a little crash course which looks at evolution from the perspective of humans. As I'm sure most of you know, modern humans came out of Africa about 100,000 years ago, and then we spread all over the world. We established human populations by 10,000 years ago, pretty much everywhere on the planet. And the first thing humans, like all other species, do when they come to a place is they protect it. They don't want anyone else coming there. And so they isolate themselves on these land masses. If you think about it, just the people in this room, every single trait in you that makes you is variable. Your height, your color, your intelligence, pretty much everything is variable. That's why we're all different. And these variations tend to get selected for by our environment. That was the principle of natural selection that Darwin, in his groundbreaking work in 1859, explained. And so in each of these genetically isolated human populations all over the world now, there was selection. Selection for living in the cold, living in the sun, living in the rainforest, living close to the water, living close to the sea, living in circumstances of aggression, and so on. If you take a simple human trait like height, we're all different heights. I'm pretty much the world average height at 170 centimeters. Some of you are much taller, some of you are much shorter. But our height, if you take it as a function of our human population, you can see it varies as a bell curve. And pretty much every trait in all animals and even in plants varies according to a curve like this. And so it was that these genetically separate human populations all over the world very soon came to look quite different. In Africa, the man on the left is from the Dinka tribe of South Sudan, and the men in that tribe, on average, are 6 foot tall. On average. Not far from him, in Central Africa, in the rainforest of the Congo, pygmy tribesmen are 4.5 feet tall. And in that same way, we diverged. In just 100,000 years, our genetically disparate populations all over the world came to look quite different. We diverged across hundreds of traits, these traits that we are so familiar with. And so if you think about the first member of any lineage of fishes that entered Sri Lanka, it had to have come only from India, because otherwise we are surrounded by ocean, freshwater fishes couldn't have got here. And so whenever sea levels were low enough, it was possible for freshwater fishes to migrate across the Pork Isthmus, this land bridge that connected us to India at various times. And when that fish got here, it found a landscape that was very complex. More than 100 river basins, mountains that went from sea level to 2,500 meters, savannas, rainforests, floodplains, the works. And as these species diversified into these various ecosystems, just like the humans did all over the world, they went on to develop genetically distinct populations. So if we take a group of fishes like the giant danios, these beautiful blue and gold fishes, we have 5 species of them in Sri Lanka. This is one that Hiranya discovered in Aranayaka. It's found only in Aranayaka on the Maoya. And he named it for the people who died in the 2016 landslide there. More than 100 people perished in that at the time he was doing his research, so he named it Memorialis for them. And this species has been isolated in the Maoya and gone on to look very different from other species of Devario, like, for example, these ones. Now, while that species is isolated in a particular river basin, these species occur across multiple river basins, but what separates them is their habitat. The one on top, Devario micronema, lives in the rainforest, in shaded streams. The one at the bottom, Devario malabaricus, lives in open habitats. And even though they might be in the same river, they've fragmented the river into an upstream, rainforested, shaded area occupied by micronema, and then malabaricus occupying the lower reaches. So evolution can separate us not into genetically distinct groups or lineages depending on hard boundaries like river basins, but even by soft boundaries like habitat. If you look at another species like these stone suckers, they are gorgeous little fishes and they're used in spas sometimes to scrape dead skin off your feet or your hands, in this case Hiranya's hand. They are recognized by one species principally in Sri Lanka. But Hiranya's work, looking at the genetics of these species, showed that there were in fact 6 very distinct lineages present. So this highlights the weakness that we have when we try to deal with conservation through the species framework, because the IUCN Red List, for example, has only species in it. But here in Sri Lanka, we find that this one species, nominal species, actually is present as 6 genetically distinct populations. Some might choose to call those species. And from studies like this, we can infer a lot more information that isn't immediately obvious. This work showed, for example, for the first time, by reading the molecules, if you will, that Gara, having come to Sri Lanka, diversified across the island. Then there was a bottleneck. They were extirpated all over the island, except in the very southeast, in a refugial population that persisted around Madara in the Ging-Nilbala basin. That was probably because there was a desiccation event. Climate change at some point, about 3 million years ago, caused their population to collapse all over the country except in the southwest of the country. The southwest of Sri Lanka is well known as a refuge now for fishes as a result of Hiranya showing time and time again that this has happened. Climate reduced our forests and our species, especially the endemics, to this southwestern corner, and now we are in a relatively wet period when they have spread around again. We know from this that that extinction event occurred. We know from this work that when Gara, these species of stone suckers, recolonized the island, they did so via the northern basins. When they got to the Mahavali Basin, to the highlands of Sri Lanka, for example, because they like highland streams, they went to the highlands through the Kala and Malbatoe, and then the Mahavali, and then up into the highlands. A similar picture happens with these stone loaches. We recognize 3 species in Sri Lanka, in the genus Schistura, but actually the genetic studies show that there are 8 very distinct, genetically distinct populations which could be recognized as species. So that is basically the way in which all these diversifications of endemic species have happened in the country. But now if you look at how did evolution take place on the broader framework of our region. You all know that we were a part of Gondwana land a long time ago, 120 million years ago, and when Gondwana fragmented, India and Sri Lanka started their journey up towards Asia across what is now the Indian Ocean. And while we were doing that, 66 million years ago, there was a catastrophe. An asteroid crashed into Earth. Not close to us, on the other side of the planet, on the Yucatan Peninsula of Mexico. It wasn't, as standards go, a big asteroid. It was about the size of Naduntivu Island, Kites Island, the little red dot you see there. That was how big it was, but it was moving at more than 70,000 kilometers an hour. And at such velocity, it had enough energy to cause untold damage to the planet. It raised so much dust into the atmosphere, sunlight was cut out, most plants died, the food chain collapsed, leading, of course, to the extinction, not just of the dinosaurs, but of about 75% of the species on the planet. As if that were not enough, shortly thereafter, there was this massive outburst of volcanism on India, in the region of what is now Mumbai or Maharashtra. More than 700,000 cubic kilometers of lava came out. It was enough, if you put all that lava on Sri Lanka, it was enough to go up to the height of Mount Everest, basically 10 kilometers up. And so when we woke up the next morning, as it were, in 65 million years ago, our landscape would have looked something like this, barren of all life, a clean slate. Very few species survived that. There were a few spiders, a few scorpions, mainly animals that live underground. But very soon, the land came to be colonized by seeds that were coming in from the air, by being carried by the wind, carried by bats, and so on. This is a photograph of Hawaii. Hawaii is one of the most isolated islands in the world, but still, plants and animals are able to reach it and then to diversify on it. Hawaii has been isolated only for about 5 million years. So Hawaii, like Sri Lanka would have had then, has no freshwater fishes. The only freshwater fishes you get in Hawaii are species that have larvae that live in the sea. We've got a few of these in Sri Lanka. This is one of them, Siziopterus. These fish live their adult life in fresh waters, but their larvae live in the sea. This is another one known as the lipstick goby, though it's the boys that wear the lipstick in this group, which also live in freshwater streams, but their larvae go down to the sea. And so, for 15 million years, Sri Lanka remained with India, isolated in the middle of the Indian Ocean, with very little life on it, certainly very little diversity. And then, about 50 million years ago, we made contact with Asia. And then there was a massive flood of species from Asia into India and down the peninsula of India into Sri Lanka at times when sea level was low enough. At that time, the region of Bengal, where this interchange of species was taking place, was roughly at where the equator is. So it was very tropical, an ever wet region. And this massive influx of new species from Asia was able to come in and colonize India. And so long as a land bridge existed to Sri Lanka, to colonize Sri Lanka. That happy situation of having this belt of rainforest leading into Sri Lanka existed till about 5 or 6 million years ago. As a result of that, we find this intimate relationship between the flora of Sri Lanka and the flora of Southeastern Asia, of Borneo, of Sumatra, of the peninsula of Malaysia. So two conditions were necessary for us to receive this gift of species. The first one was that sea level had to be low enough for there to exist a land bridge between us and India. Now, such a sea level lowering happened during the last glacial period. Sea level was 140 meters, 120 meters, 400 feet below its present level. The land bridge to India, 20,000 years ago, very short time, humans were already here by then, was 140 kilometers wide. It was as wide as Mannar to Trincomalee, leading all the way to the peninsula of India. And the second thing was we needed to have a wet climate in the region of the land bridge. Now you know that Mannar is the driest place in the country. It's very low rainfall, and that low rainfall has lasted for pretty much all of the last 5 million years. But before then, species associated with rainfall could come into Sri Lanka. And then, about 30 million years ago, something else happened. The region of Bengal, because now the Himalayas had been rising, the southeast and northwest monsoons had been, northeast and southwest monsoons had become increasingly more powerful, and the climate was becoming more and more seasonal. They were getting a long, dry season in the region of Bengal, what is now Bengal. And so there was a climatic barrier between India and Southeast Asia. That great interchange of species between Southeast Asia and India came to an end. India was then isolated on the Indus Valley, even now, which is very dry. Pakistan, Gujarat, Iran, Afghanistan, and that region, notoriously dry. And to the north, India was cut off by the Himalayas. Now India was, as far as biology was concerned, an island. Meanwhile, the south of Sri Lanka still maintained, even at the worst of times, rainforest. So we had this dichotomy when we had rainforest across the Pork Strait, the Pork Isthmus, the land bridge. Species were able to come into Sri Lanka. And when we had this dry Pork Strait region, species could no longer migrate. And Sri Lanka benefited from another characteristic. This is our driest month, and you can see that we are lying at this northern fringe of this belt of rainfall that we have right around the equator, called the Intertropical Convergence Zone. This is our driest month. And then, as the year proceeds, you can see this belt of rainfall moves north with the southwest monsoon and then pushed south again by the northeast monsoon. So we get rain not only during the monsoons, but during the intermonsoons. That's why the southwest of Sri Lanka is such a wonderful place for biodiversity and has no real dry months. I'll come to that later. So what were the consequences? We talked about those species that came across India from Southeast Asia, and amongst them was this group. This is an endemic Belontia signata. In Sinhala, we call it the Thalkossa. When Hiranya did the genetics, this wasn't Hiranya, this was Hiranya's boss, who was already working on this, Dr. Lucas Ruber in Switzerland. He found that the closest relatives of Belontia signata were in fact a Southeast Asian group. Now, Belontia doesn't occur anywhere else in between, not in India, Bangladesh, Myanmar, Thailand. You have to get to the southern region of Malaysia before you find it again. So the closest relative of the Sri Lankan group is in Southeast Asia. Now, this is not an exceptional case. This is the norm. When we look at plants, for example, we find that many of these wonderful endemics that we have, like Axinandra, have closest relatives nowhere else in Asia, but in those Southeast Asian islands. That is, if you like, where the mother lode of our biodiversity came from. Groups like these wonderful lizards we have in Sri Lanka, Ceratophora, Cofortis, and the other one, his name eludes me, Lyriocephalus. They all have no relatives on India. Their closest relatives are again in those Southeast Asian islands. So that was our Southeast Asian link. But then we have an even more fascinating link to the West. This photograph shows the Asoka barb and the black-line barb. Both of these belong to the genus Cystromus. And Sri Lanka has the greatest diversity of Cystromus that we know about right now. These species have been diversifying on Sri Lanka for a long time. Now cast your mind back to the fact that we met up with Asia 50 million years ago, and if you look at that picture, you'll see that Africa is an island. It's not connected to any other landmass. Then about 30 million years ago, Africa finally connected with Europe, with West Asia, across the Arabian Peninsula. Now there was a land bridge, not just from Sri Lanka to India and India to Southeast Asia, but also effectively from Sri Lanka to Africa, doing that circuitous route. And one ancestral species of Cystromus was able to make the journey into Africa, into the equatorial rainforests of Central Africa. And there it diversified into more than 200 species which are known as the African small barbs. There's one picture there of an African small barb. This is after 30 million years of evolution. And as you can see from the picture of the black line barb at the bottom of the pictures on the right, there is a remarkable resemblance even all after the passage of so much time. So we made these lovely biotic connections, even as evolution progressed through that period. Now I want to leave that kind of evolution to one side and start looking at another kind of evolution. What I think of as the Dawkinsia playbook. This is a specimen of Dawkinsia filamentosa, a really common fish in Sri Lanka. It's beautiful and it's one of our most common fishes. Everywhere in the lowlands of Sri Lanka, every river, every tank in the dry zone contains this. They're not always in such beautiful color. This is how you're likely to find them. But what is remarkable about these fish is that they have these long trailing filaments from the fin on top of the fish, the dorsal fin. These dorsal fin filaments, which are absent in their females. We have two species in Sri Lanka, this one, which is as common as dirt, and another one, which is quite special, which is found only in the knuckles, discovered by Dr. Ranil Senanayake in the late 1970s, called Dawkinsia srilankensis. This is endemic to the knuckles, the rivers flowing north from the knuckles. And when Hiranya did the genetics for this, he was in for a surprise. He found that the closest relative of the Dawkinsia in the Knuckles Hills was a species in India, in southeastern India, in the Tamiraparani River. This was a wonderful discovery because it showed that there had been a hydrological link between Sri Lanka and India at that time. And then from the timings of the molecular clock, you're able to show that that connection existed about 3 million years ago. So this is how we are unraveling our history as an island from molecules. How did this happen? How did an ancestral species of Dawkinsia come to Sri Lanka? Obviously, it came through the Pork Strait. But this means we must have had rivers that connected the knuckles to the Southeast Indian rivers. And indeed, when Indian scientists have been doing the hydrogeology of this region, they show that that was indeed the case. So you have the rivers draining out of Sri Lanka into the Pork Strait region, the Malbatu and Kala Oyas, connecting with the Vaigai and the Vaipal and the Tamiraparani drainages of southeastern India. All these rivers flowing into the Pork Isthmus, the land bridge, and becoming confluent there, and then flowing into the Arabian Sea from there. Not only that, remember the connection was with the Kala and Malbatu Oyas. But the Kala and Malbatu today don't drain the Knuckles Hills. The Knuckles drains entirely into the Mahavalley. But Pethiyagoda, in a completely different context, almost a century ago, predicted that the knuckles had at some time drained into the Kala Malbatu drainages. And here we have evidence that that actually happened. At some time in the past, around 3 million years ago, the knuckles was connected to the Kala Malbatu drainage and not to the Mahaveli drainage. Now, based on this, we can make a few hypothetical predictions because there are other endemic fishes in the knuckles. For example, Martinstein's Cystomus and Labeo fisheri. It is entirely possible one day in the future that we will discover their closest relatives, because they have no close relatives in Sri Lanka. We will discover their closest relatives in southeastern India, because we have now discovered that that hydrological link existed. When we first described this genus Dawkinsia in 2012, we chose to name it after Richard Dawkins, who is probably the greatest evolutionary philosopher since Darwin. There have been many, so I shouldn't say greatest, but a great evolutionary philosopher. He especially was relevant to this because the male and female of Dawkinsias are so different by having those trailing dorsal fin filaments in the males. Darwin himself referred to this in a second book he wrote in 1871, The Descent of Man, the subtitle of which was Selection in Relation to Sex. Now, his first book in 1859 was about natural selection. Now he was talking about sexual selection. Sexual selection is familiar to everybody, even if you haven't thought about it. There are things that women look for in men and things that men look for in women. Women, for example, like men who are taller. This is universally true. And so human beings would be selected for male tallness. Men like women for other attributes. And those attributes naturally become more emphasized in women. That's how sexual selection works. So is that what's going on in Dawkinsia? Is that why the males have these trailing filaments from their dorsal fin and the females don't do it? Do females happen to prefer males who have got long dorsal filaments? No one has done the science up to now. We don't know what the answer to this question is. But there's a PhD waiting to be done on this because this is not a trivial question. Now, when I told you about this situation, I'm sure a lot of you thought, that sounds a bit like the peacock. Because the male and female peafowl are so dramatically different. And we think of the male peafowl as being something that likes to display itself, and the females obviously seem to like something about the display. The males have this long train behind them, the colorful ocelli on their plumage that must be attractive to females. I mean, men feel like this all the time. They feel the need to go to the gym and put on upper body muscle, for example, to make themselves more attractive to women, even if they don't consciously think about it like that. So for 120 years, from 1871 to 1991, this was the received wisdom taught in every university, that the peacock was this wonderful example of sexual selection. Finally, in 1991, somebody subjected this to experiment. They looked at whether female peafowl, whether peahens actually preferred peacocks with longer tails and more eye spots on their tails. And the experiments confirmed the result. It seemed a wonderful proof that Darwin had been right all along. But then people began to question this, because after all, as a peacock ages, its tail gets a bit longer. Were the peahens choosing peacocks because of their longer tails, or because the peacocks were older and more mature? And that turned out to be the case. It was shown that peahens don't prefer peacocks with more elaborate trains. If you like, think about it like this. There's no woman in the world who thinks of this kid as sexually attractive. He's a 13-year-old sexually mature boy, but he's not attractive to women. 5 years later, he's another proposition. So that's what was happening, apparently, with the peahens. They wanted peacocks that were not just ready to mate, but had some maturity about them. Because look at the advantage from the evolutionary point of view. If you had proved not that you were sexually mature, but that you had been sexually mature and survived for 5 years, you obviously had proved that you were worth selecting, that you could survive. And that's what every mother wants for her children, is the ability to survive. That's why she wants the genes from them. And so, as Darwin wrote, a peacock's tail in all its beauty would be a disadvantage to him in the struggle for existence, if it were not the case that peahens were particularly attracted to his plumage. The received wisdom was that by having this expensive trait, like a Ferrari in the case of men who want to show that they're worth selecting, the peacock was taking this massive train behind him because it would make him less able to escape from predators, less able to run, less able to fly, less able to escape. But then the science showed that that wasn't the case either. Peacocks, despite their long train, the males can run perfectly well. They can fly perfectly well. So what on earth was going on here? We've all seen the peacock display, raising its feathers like this, and then giving this lovely shudder. And once they do that, if the sun's coming from there, they turn themselves exactly at 45 degrees to the sun, to the azimuth of the sun. And they hold that pose for a while. And this shiver is something that is very important to them. And people started thinking, is there something relevant in the whole shiver thing? I can't do it. I'm not a good dancer. But there are those of you I know who can. I've seen you. And as scientists do, they recorded the shiver and found that it was exactly at 26 cycles per second, 26 hertz. 26 hertz is about the lowest note on a piano, A0. If you play the leftmost note, so you can imagine what that sounds like. That's the resonant frequency of the peacock's shiver. And then people noticed, hang on a minute, males and females of peacocks are so different. They've got all different ornaments, but there's something in common. Peacocks have this little coronet of feathers on their heads, and what do you know, the peahens have the same thing. So they started shaking that about and recording the resonance frequency of that clump of feathers on the head, and turned out that that was 26 hertz exactly as well. So it looked like the coronet is something that can be used as a listening device to help its hearing to hear those low infrasounds from the shudder. Now, both males and females are listening. So is the peacock displaying only to the females, or is he also sending a message to the males? And very soon, the science was in. Peacocks and peahens are sensitive to ultrasound, and they use the crest for detecting ultrasound. Now, that's where the science stands today. People who study peacocks in a state of complete confusion as to what this hugely different morphology between male and female actually mean. And I'm showing you this map because, as we know, peacocks are not just common to Sri Lanka, but they're now a pest in Sri Lanka and in India. But none of these studies has been done in Sri Lanka. This is fascinatingly interesting stuff. And this animal is sitting on our doorstep, literally. But all these studies have been done in North America, in Japan, in Europe, and so on. So if the peacock, the best known example of this in the world, is so difficult to understand, how much more difficult potentially is Dawkinsia? Dawkinsia, unlike peacocks, have a relatively short time to maturity. They can be bred very easily, and they have been studied in the context of species like the swordtail experimentally. Here again, male swordtails have the sword on their tail and females don't. But when we look at the swordtail, if we artificially stick a tail, for example, on a platy, it becomes very attractive to female platies. So it seems that size is all that this sword is indicating. And that gives us a new perspective on how evolution might be working in that case I showed you earlier, the Dinka tribesmen of southern Sudan. Are they tall because something in their environment is selecting for tallness, like the ability to evaporate sweat, or run long distances, or have more stamina? You all know that it's always southern Ethiopians and northern Kenyans who win the marathon every year. There's got to be something in their genes. Or is it that Dinka ladies are choosing progressively taller men? So even when it comes to humans, these are really tough questions to understand. Another example from our fishes, this is Schismatogobius deraniagalai, a species that I discovered in 1989 with my wife, who's in the audience today, in a little stream, the Vioya, near Yatiantota. An unusual fish, again, its larvae go to the sea to breed, and it has this huge gape. The males have an enormous mouth. You can see the size of the mouth in this male here, and here's a female with a much smaller mouth. Now, Hiranya recorded them. These fish are quite rare, but he recorded them underwater. And this is a male going in search of a female, and when it comes to the female, it opens its mouth bright orange, tambili orange on the inside. Here it is, displaying to the female, and then gives the female a nasty love bite. We don't know whether that's a sign of aggression or affection. Somebody in the audience should do the research to find out. I'll just quickly show you that again. Coming up to the female, displaying, and then a little bite. These little aberrations amongst fishes make them strangely human in many ways and remind us that we ourselves descended from fish. There is an inner fish in us. Moving on. We see now in the news quite often that this story, this narrative of gender fluidity and sex role reversal in the West, nobody knows whether they're boys or girls. We are told that sex is a spectrum, for example, but of course it isn't. There was even a documentary 2 years ago by this guy, Matt Walsh, asking, what is a woman? Best widely watched documentary all over the world, because now it's politically incorrect to say that you can recognize a woman. A woman is anyone who says they're a woman, not anyone who's born that way. But of course, this is a nonsensical argument, because we all know that in mammals, a female is chosen by her ability to nurse her young. But that's not true for the other groups of vertebrates. The greater painted snipe. Here, the bird on the left is actually a female. The females are bigger and more brightly colored than the males. And the sex roles have been reversed. The male builds a little nest and he just sits there in the role that has been popularized for females, kind of passive role. The female comes there and she displays to him, and if he likes her, he'll mate with her and she'll deposit 4 eggs in his nest and move on. The male is then left to do all the housekeeping. He's got to incubate the eggs, and when the young are born, he's got to play mummy. The female, for her part, will go around and mate with up to 10 different males, depositing eggs in all their nests, like a male would normally go mating with females. And what do you know? There's a fish that does exactly that, the ocellated pipefish. This is not a common fish, but we find it in our streams, in the lowlands, in the rainforest streams. And here, the female develops the eggs. She goes up to the male, they mate, and then she passes the whole mat of eggs to the male. The picture at the bottom is a male showing the underside, and you can see he's got a long furrow along his underside. She passes the eggs into that furrow, and he keeps them there and incubates them. She goes off in search of another male, and the male brings up the babies until they are independent. Perhaps the nicest of these fish is this guy, a very common fish in Negombo Lagoon, for example, or in Bolgoda. the cichlids. They came to us from Madagascar. We don't exactly know when or how. But we have two species in Sri Lanka. The one on the left is the green chromide, and the one on the right is the orange chromide. The green chromides are vegetarian. They're about a foot long at most. And the orange chromides are about 1/3 as big. And they've got really interesting stories to tell about their behavior. When the orange chromides are breeding, they change color to like the fish on the right, they get these black markings. So this is a non-breeding orange chromide, and here's a breeding one with these black markings. And in the foreground, in the lower foreground, you can just about see the black dots, which are its babies. You can see them more clearly here. And when they have young, the parents look after the young, and as they go around looking for fish to eat, they tend their young right through the course of the day. So here's a pair of orange chromides taking their young around, going on their daily round. As you can see, they're very vigilant, always turning around and checking on whether predators might be approaching. And in between this, they've got to do their hunting so that they can stay alive, because they're piscivorous and they need fish to eat. And then occasionally, you'll find that a predator whizzes by and takes a few of the young, like there he goes. And so a few of their young get eaten. But they spend pretty much all their life doing that. Green chromides, on the other hand, are much more gentle fishes. They lay their eggs on hard objects like this underground. This is in an aquarium. And then the adults will fan the eggs and look after the eggs till they hatch. And the funny thing in these fish is that even the aunts and uncles of these babies come and help, just like in a human family, to look after the young, to fan the eggs. And when the fry are hatched, the parents don't feed while they're looking after the babies. The aunts and uncles move around with them, helping to look after. Here's a cluster of young green chromides, and you can see the parents looking after them. Now, a problem the green chromides have is that they get parasite infestations and fungal infestations quite often. Now, when mammals have this problem, even humans, we do this. We groom each other, pick out lice and so on. And what do you know? With the orange and the green chromides, the same thing happens. The green chromides, when they have parasites, they adopt this really bizarre posture of pointing their head up. And then the orange chromide comes and cleans the parasites off it. What's in it for the orange chromide? The orange chromide does this because it then gets to approach green chromides and eat their young. The orange chromides are really complicated fishes. So it's almost as if the orange chromides are doing what we do with chickens. We rear a chicken, we feed it, we look after it, and then we eat its young. That is pretty much what the orange chromide is doing to the green chromide. The orange chromides even do something more bizarre. They kidnap the babies of other orange chromides. Say an orange chromide is going around with 100 young. It'll go and get 50 young kidnapped from one of its neighbors and joined into its group of babies. Because what's in it for the orange chromide? Now, if half the young with it get eaten by a predator, the chances that its own young will survive are greater, because half the guys that are going to get eaten are somebody else's kids. So they're really complicated fish. You might wonder, why don't green chromides clean each other? The reason seems to be that they're bigger, they've got bigger mouths, and they've got these very flat teeth, with which I presume it's more difficult to grab these bits of parasites and so on. The green chromides, because they eat fish, have got much sharper teeth. You know, the point we hope to make, Hiranya and I, in telling you these stories, is to urge ourselves not to think about biodiversity and conservation in terms of this static photograph we take called species. Evolution is about processes. It's not about species. These wonderful processes are going on around us. And that is what we need to conserve. Conserving species is just one small element of the processes that create the species. It's not enough to protect the species. And this message becomes even more important as we approach a period in which we are looking at enormous stresses to biodiversity through this wild card, the joker that is being played entirely because of our fault as human beings of precipitating very rapid climate change. Sri Lanka's southwestern wet zone is a storehouse of biodiversity. We all know that. It's a storehouse of biodiversity because we've got this really special climate. There is no place in the whole of South Asia that has got a climate like the southwest of Sri Lanka. Look at the difference within Sri Lanka itself of rainfall. The top is the dry zone, Jaffna, Mannar, Vavuniya, Trincoe, Anuradhapura. They all have long dark blue months where average rainfall is less than 100 millimeters a month. And then look at the wet zone at the bottom. There are almost no consecutive months in which rainfall is less than 100 millimeters. We have this ever wet per humid climate. It is so important that there is nowhere else between equatorial Africa and peninsular Malaysia with a climate like the southwest of Sri Lanka. This is really special. It's a big deal. I've done a lot of work in Kerala. This is Kerala's second biggest river, the Bharatapura. And the Bharatapura river flow looks like this. Lots of flow in the second half of the year and almost no flow in the first half. This is what the river looks like during the dry season. You compare that with a similar river in Sri Lanka, the Kalani, with which we are all familiar. There's no such fall in flow even during the dry season. The Kalani is always flowing. When you look at the difference that this little subtle change in climate makes, just 300 or 400 kilometers to the north of us in Kerala, it still has rainforest, but the aquatic biodiversity has come to be gravely threatened. So the message we wanted to give you, the two of us, is that what we need to think about, especially as a result of the discoveries that Hiranya has made through genetic analysis of demonstrating that we've had these extinction events in the not too distant past, is that these events will come again as climate intensifies. And we have to spend as much effort, as much brain power as we can, because now conservation is no longer a pastime. Conservation is now rocket science. We have to invest our best brains and as much intelligence as we can muster to address these challenges as we go forward. Thank you very much.
Thank you, Rohan, for that brilliant 50 minutes of awesome information. We have about 15 minutes for a few questions. Again, I'm mentioning it's questions and not statements and opinions. Please ask your specific questions and Rohan will be happy to answer.
Yes, my first question is that cichlids found only in Sri Lanka and India, the three species, but how they came from with the separation from Africa and Madagascar, or from the other way around, like that Barb you told in the earlier. Which way it came to Sri Lanka?
We don't know yet. That's an excellent question, actually. But the short answer is we don't know, because the molecular clocks show that the Sri Lankan and the Madagascan cichlids, their closest relatives are on Madagascar, a genus called Paratroplus. And on Madagascar, our cichlids and theirs, according to the molecular clock, diverged only about 30 million years ago. That is too recent, because Sri Lanka and Madagascar diverged from as being a landmass about 90 million years ago, 88 million years ago. So the problem might be that our molecular clocks are badly calibrated, because to calibrate them you need fossils that can be dated, and the problem could be that we don't have fossils. So I think the short answer is we don't know. That's work to be done in the future to discover how that connection came. Hiranya is also online, I believe, and can take questions if anyone's got any for him. One question you can ask him is how he plans to find a job in Sri Lanka. Spencer?
Yes, Hiranya, come
We can wrap up, I think. No more questions. Nimi.
Okay, Rohan, if the species obviously is no longer the target, what's in it, okay, for Sri Lanka's conservation? Because I presume that both you and Hiranya think that the evolutionary selective forces are still operating on these populations, okay? All right. So given that fact that we cannot change climate, we cannot, do anything about climate change at least for the next 50 or 100 years, how then should conservation be? Because you're talking about the processes that are driving. Thank you.
Hiranya, do you want to take that?
Yeah, sure. Can you hear me well?
No, we can't hear him. All right, so I'll start answering while he gets his microphone working. So I think, Nimi, it's only now that we are able to recognize these genetically distinct populations that might be morphologically quite similar. Like in the case of Schistura, these loaches, 8 morphologically very similar populations which are genetically distinct. But now, for more and more species, we are developing this information. I think for the amphibians, we've got really good information. People like Chanel Vikramanayaka have been working on the gamete lizards and so on. And so we are getting a feel for how these populations can be distinct, even though we are still treating them as one species. And I think the time has come for us to abandon this Linnaean concept of species that is now 350 years old. I shouldn't be saying that because I'm part of that establishment. But actually, for conservation purposes, it's archaic. We need to think about genetically distinct populations. Hiranya, can you be heard now?
How about now? Can you hear me?
Nope. So I hope that was the answer that he would have given. Effective. Hiranya, can you say something?
I can say something. If I
Right, now we can hear you. Did you hear Professor Pallavasta's question?
Oh. Can you repeat the question again, Ron?
Start looking to conserve genetically distinct populations. And if so, how?
Yeah. I think, of course, when it comes to conservation, having a species concept is really important. It's like a focal point, but the thing is to understand the processes itself, I think adds up on the value of conserving the whole system rather than just pinpointing, selecting a species. But I think, as Rohan said, it's the ideas of species concepts there are so many species concepts, and it's like when you study a system and when you go more and more deeper into a group of I don't know, family or a genus, like the spectrum where you draw the line is not so straightforward as like what you would like to see, like if this morphological character is different, we call it a species A versus species B. It's not so straightforward when we analyze more data and more at the population level. So I think that's what like all other studies, like what work of Chanel as well on the lizards, that show that the population level genetic studies adds more on top of what we just call species at a conservation point of view, I guess.
Thank you. Good.
Rohan, thank you for an excellent lecture. I just want to pick up on Professor Pallevat's point. So I've got two related questions, and they're both to do with the interplay of conservation and phylogenetics. So firstly, with the Gara, the 7 clades, so from a conservation perspective, there'll be some pressure on the people who are doing taxonomic work to describe 6 or 7 species so that you conserve them. So it's going back to Hiranya's comment about there are multiple species concepts, but whatever the concept, species becomes important as a conservation unit. So the first question is how you deal with that pressure, do you hold back, or do you say, well, if you don't describe this as a species, we could lose that, and, is it a species or is it a variation or whatever? Then related to that, are there any ancient lineages that have, where the descendants are there in Sri Lanka, but have died out elsewhere, so that conservation-wise, because we want to preserve that phylogenetic history, there are things in Sri Lanka which warrant special conservation concern?
Hiranya?
Yeah, I think the first question is, different.
I can't hear you.
Can you hear me now? Hello. I'm sorry, but I don't know what's going on.
We got you.
Okay. Can you hear me, I guess? Can you hear me?
We can hear you, but did you hear
Okay, cool. In the case of Gara, for example, yes, like now we have 1 species, but now the genetic data finds out, okay, we have 8 distinct lineages. So like then the question is just for this conservation, do you need just a species name to conserve? Or I think I would rather say you don't really necessarily need a species to conserve. Even you can and identify the whole different 8 distinct genetic lineages without giving a name. And for example, in the United States, like the concept of the evolutionary significant units is quite routinely used in conservation, where they identified even within a given species, you have different genetically distinct populations, and they give different emphasis when it comes to conservation action. So I don't think necessarily you need to have a name to conserve. Even within a species, if you recognize different genetic populations, for the action, conservation action, you can have different priorities, I think.
Sure. Can I add something to that?
Yes, please.
Is that we are mixing water across our basins quite routinely now. The Umaoya, the Mahavali, which has been separated from the southern basins, from Kirindi and Manik and so on, these waters are now getting mixed up because of trans-basin diversions. And so fish go with the flow as well. And eventually we are going to be diluting these populations and mixing them up because of these human interventions. I suppose that those are inevitable, but this is something that ex situ conservation, conserving these lineages, even in zoos or something, until we can find out what to do to preserve this diversity, is worth thinking about. The Dehibala Zoo has made some small inroads into conserving a couple of species, and these are at the very beginning of their projects. They've been doing it for a few years, but there's a lot of science that's yet to come, but they're trying to do it properly by doing genetic analysis of their populations, by taking quarantine restrictions and so on. Your second question, Gihan, I think Hiranya didn't hear, and I know he'll disagree with me because we thought of talking about this, but he cancelled it from the talk, was these lineages that might be unique in Sri Lanka and very separate from everything else. Tithya the little red cherry barb is a good example. No real idea of what its closest relatives are, apart from, we've got Puntis bimaculatus, so what is now another genus. But we've got these genera like Rasboroides, what's the common word for Rasboroides? I forget. I don't think in terms of common names. But anyway, Rasboroides, these tiny little Rasboras, brilliant colors, the Halmaldandia in Sinhala. We don't really know what the closest relatives of these are in terms of Sri Lanka. There's no close relatives in Sri Lanka. There's no really close relatives in India. And Hiranya's work, I think, will show close relatives in Southeast Asia. That's as much as I dare say, because this is an area of dispute between him and me. So what do you like to add, Hiranya?
I think for that example, like Belontia would be a good example.
Nope, hang on, wait for the sound. Keep saying something, because then they'll hear you. They'll hear.
Dang, ඇහෙනවද? තාම නැහැ. දැන් කියනවා ඇහෙනවද දැන්? Okay. So I mean, Belontia would be a good example, right? Like you have Southeast Asian lineage, and then you have a Sri Lankan lineage. I mean, it is possible Belontia lived in the past in Indian subcontinent, and then it went extinct there as well. That is a possible hypothesis. So the fact that we have two distinct lineages pretty far away geographically, one species in Southeast Asia and one in Sri Lanka only, that's a pretty, I think, a remarkable example. And another example, maybe I see Kumudu and Dinosaurus, they are maybe like ours, for example. Maybe that's a pretty ancient lineage that is, I don't know, the most related would be in Madagascar or somewhere, but I mean, yeah, sure.
So much for your talk. I would like also to ask about marine conservation. I want to talk about the international conferences such as the 30 by 30 goals of the UN to protect 30% of terrestrial and marine landscapes across the world, and then also the SDG number 14, life below water. For the 30% of marine protected areas by 2030, Sri Lanka is only at 0.1%. And I'd like to ask your perspective in terms of conservation, of why you think Sri Lanka is struggling so much to meet those international goals that we've signed on to, and then how we can best go forward.
I'm not the best person to answer this question because I'm not a marine person. I think Asha was here somewhere a minute ago. And she's probably the right person to answer that kind of question, not me.
Dr. Malik, would you?
He has no question.
Anyone else would like to answer that question? I'll connect you with Professor Sevandi. Yeah, fine.
Yeah, I think I'm not a great fan of any of the UN conventions that relate to biodiversity, including the CBD. So this is a whole other ball game. I'm very cynical about these international conventions personally. I could go on and on to tell you why, but I don't think I want to bore everybody about this.
Okay, maybe we can talk later. I'm very interested too.
I basically see it as white people dictating to the brown people all over again. That's why I think we should just tell them to buzz off.
Okay, on that high note, could I call President Graham to hand over a gift and say a few words?
Thank you, President. So, Rohan and Hiranya, thank you so much for that brilliant presentation on the very arcane subject that very few of us have in the conservation community have too much knowledge on. You've also dropped or parachuted into the laps of the hands of the conservation community the concept or the paradigm that conservation of species could be looked at also through the lens of genetics and not only species only. So with that thought, I'm sure you've engaged the scientists and the community enough to come back and look at things in a different manner. So thank you very much for this very engaging evening.